In this video, we will discuss about some of the induced proximity between two cells and how they can be used, or this phenomena can be leveraged to improve the immune system response against the cancer cells. So first one is about this T cells and the target cells. The target cells can be considered as cancer cells or tumor cells. So in this case, how the cell proximity works is that the T cells will able to interact with the major histocompatibility complex and with the presence of the peptide. So if these T cells already has these informations about the peptide, and when it interacts with the MHC and it is able to recognize that peptide, what it will do is it will recruit other T cells, and those phenomena then together will trigger the activations of the T cells, maybe that it will release the cytokines that will then target these T cells and kill those T cells. This is one of the examples. You can understand about this, how this T cell cytotoxicity work in this case in one of our videos where I have described about this in a detail. So let us now focus on the other part. So in this case, the bifunctional antibodies, what it will does is it is also known as T cells engager. What it will does is it will brings the cancer cells and the T cells close to each other. So on one hand it will has the antigens of this cancer cell, cancer antigens. One hand it has the receptors that can bind to the cancer antigens on the target cells, and other hand it has the receptors that can bind to the CD3 epsilon ligand that are present on the T cells, or receptors that are present on the CD3 epsilon receptors that are present on the T cells, and thereby bringing these target cells and T cells together. And in this case, when the target cells comes close to the T cells, then these T cells will be able to recognize these tumor cells or the target cells, and will trigger the immune response that will finally leads to the killing of these target cells. So in this case, the strategy is different from the panel A, in terms of that in the panel A, the T cells already has the informations about the peptide that are present on the target cells, and when it interacts with the major histocompatibility complex, it will check whether that same peptide is present, and if it is present, then it will generate the cell killing response. In this case, the T cells engagers will bring the target cells to the T cells, and that close in the proximity will help the T cells to recognize that these target cells are harmful to the body, and that will lead to this cell killing response. In the third case is taking advantage of this trogocytosis. And trogocytosis means that when the two cells are close in proximity to each other, then it will cause the exchange of the intercellular membranes, and these things they have also observed in the cancer cells. So they have leveraged these properties to generate the antibodies, thereby bringing these target cells and B cells close to each other in a way that through the generation of this trogo TAC, that is based on the trogocytosis. In this case, they use two bispecific trogocytosis. In this case, on the one hand they will have the antibodies that can target proteins on the target cells, and on the other hand they have these glycan structures that can bind to the B cells, that is CD22 receptors on the B cells. So this trogocytosis phenomena can be used to bring this target cells close to the B cells, so that the target proteins can go to the B cells, and then B cells can generate antibodies against that target proteins that are predominantly present on the target cells only. Thereby, that antibodies that are generated by the B cells will target this target cells or the tumor cells. So in this way, this is some of the three examples where induced proximity between two cells could enhance the immune response. And if you need to understand more about their functioning in detail, you can go through the reference that has been provided in the description. That's all for this video. Thank you for your time.